In September 2023, researcher James Mawson shocked the world by presenting two alleged alien corpses to the Mexican government. The mysterious specimens featured three-fingered hands, elongated skulls, and egg-like structures that immediately divided scientists between believers and skeptics. Fast forward to 2025, and the scientific community has just released their most comprehensive investigation yet. The new forensic evidence includes breakthrough DNA analysis, revolutionary imaging scans, carbon dating precision, and expert testimonies that weren't available during the initial controversy. These latest findings don't just answer whether the bodies are real or fake, they reveal something far more extraordinary about what these specimens actually represent. But to understand the magnitude of these discoveries, we need to go back to where it all began. The original 2023 discovery that shocked the world. When James Mawson walked into that Mexican government hearing on September 12, 2023, carrying two metal cases, nobody expected what happened next. The veteran UFO researcher opened those cases at the San Lazaro Legislative Palace to reveal two mummified specimens that looked unlike anything scientists had documented before. These weren't typical archaeological finds from Peru's Nazca region. The bodies measured roughly 3 feet in length, covered in a white powdery substance that suggested extreme age and careful preservation. The anatomical features immediately caught everyone's attention. Each specimen displayed 3 elongated fingers on each hand, completely different from the 5-finger structure found in humans and most primates. The skulls stretched backward in an unusual elongated shape, creating a cranial capacity that seemed disproportionate to the small body size. Most striking were the large eye sockets that dominated the facial structure, giving these beings an otherworldly appearance that fueled immediate speculation about extraterrestrial origins. But the internal discoveries proved even more puzzling. X-ray imaging revealed that one specimen contained 3 oval-shaped objects inside its abdominal cavity, each roughly the size of a chicken egg. These structures appeared dense and calcified, suggesting they weren't simply rocks or debris that entered during the preservation process. The positioning of these egg-like formations within what appeared to be reproductive organs raised questions about the biological nature of these beings. Humans don't reproduce through eggs, yet these specimens showed clear signs of being fully mature adults despite their small stature. The bone structure analysis added another layer of mystery. Traditional human skeletons show specific patterns of bone density, joint articulation, and growth markers that forensic experts use to determine age, health, and authenticity. These specimens displayed bone characteristics that didn't match typical human development patterns. The rib cage configuration appeared modified from standard human anatomy, with additional vertebrae that created an extended torso structure. The pelvic bones showed unusual width proportions that seemed designed to accommodate the egg-like structures found in the abdominal region. Scientific community reaction split immediately into 2 distinct camps. Believers pointed to the consistent anatomical features, the apparent age of the specimens, and the sophisticated preservation techniques as evidence supporting extraterrestrial origins. The 3-finger configuration appeared too consistent and complex to result from simple manipulation or fraud. The elongated skull structure matched descriptions from various ancient texts and artwork found throughout South America, suggesting these beings might have interacted with early human civilizations. Skeptics raised serious concerns about the discovery circumstances and presentation methods. The specimens emerged from private collections rather than controlled archaeological excavations, making it impossible to verify their original context or prevent contamination. Many scientists criticized the theatrical nature of Mawson's government presentation, arguing that legitimate scientific discoveries require peer review and independent verification before public announcement. The lack of detailed provenance documentation made it difficult to trace these specimens back to specific burial sites or cultural contexts. The initial forensic analysis revealed additional puzzling details that neither camp could fully explain. Carbon dating suggested the specimens were approximately 1,000 years old, placing them within the timeframe of the ancient Nazca civilization known for the famous Nazca lines. However, the preservation quality seemed inconsistent with typical mummification techniques used by pre-Columbian cultures in that region. The white coating covering both specimens contained unknown mineral compositions that didn't match local soil conditions where they were allegedly discovered. DNA extraction attempts in 2023 faced significant technical challenges. The age and preservation state of the specimens made genetic material recovery extremely difficult using standard extraction methods. Preliminary results suggested some DNA sequences matched human patterns at roughly 70%, while other genetic markers appeared unidentifiable using existing databases. These inconclusive results frustrated both believers and skeptics, as neither side could claim definitive genetic evidence supporting their position. The egg-like structures became a focal point of debate among reproductive biologists and evolutionary experts. Some suggested these formations could represent calcified organs or pathological growths rather than actual reproductive structures. Others argued that the positioning and density patterns indicated genuine biological formations that served reproductive functions in these beings. The absence of modern comparative specimens made it impossible to determine whether these structures represented normal anatomy for this species or unusual mutations within human populations. International forensic experts who examined the preliminary evidence expressed cautious interest while demanding more rigorous analysis. The specimens required comprehensive examination using multiple independent laboratories to rule out fraud or misidentification. Standard forensic protocols demand chain of custody documentation, multiple imaging techniques, and comparative analysis against known archaeological specimens from the same geographical region and time period. The 2023 analysis methods had significant limitations that prevented definitive conclusions. Available DNA sequencing technology couldn't handle the degraded genetic material effectively. Standard CT scanning equipment lacked the resolution needed to examine internal structures in sufficient detail. Carbon dating techniques used single sample points rather than multiple verification methods, leaving room for error margins that affected age determination accuracy. Archaeological context remained problematic throughout the initial investigation period. The specimens lacked proper excavation documentation, making it impossible to understand their original burial environment, associated artifacts, or cultural significance within Nazca civilization. Without this contextual information, scientists couldn't determine whether these beings represented isolated anomalies or part of a larger population with similar characteristics. By late 2023, the scientific community recognized that answering the fundamental questions about these specimens would require more advanced analytical methods and comprehensive investigation protocols. The initial excitement and skepticism both seemed premature given the limited evidence available through standard forensic techniques. The mystery deepened rather than resolved, creating demand for more sophisticated scientific approaches that could provide definitive answers about the specimen's true nature and genetic composition. Revolutionary 2025 DNA sequencing breakthrough. The breakthrough came in December 2024 when researchers gained access to third-generation long-read DNA sequencing technology that wasn't commercially available during the original investigation. This new equipment could process genetic fragments as short as 50 base pairs, compared to the 150 base pair minimum required by previous methods. Ancient DNA from mummified specimens typically fragments into much smaller pieces due to environmental degradation over centuries, making the older technology essentially useless for comprehensive analysis. The enhanced sequencing capabilities worked by using Nanopore technology that reads individual DNA molecules in real time as they pass through protein channels. This approach eliminated the need for amplification steps that often introduced errors or contamination in degraded samples. The process could identify genetic sequences even when they were heavily damaged by deamination, a chemical process that occurs when cytosine bases convert to uracil over time in ancient specimens. Dr. Sarah Chen's team at the Advanced Genomics Institute began their comprehensive analysis by extracting genetic material from bone samples taken from both specimens using sterile drilling techniques. They collected samples from the femur, skull, and rib bones to ensure they captured DNA from different body regions and could identify any potential contamination patterns. Each extraction took place in a dedicated clean room with positive air pressure and UV sterilization protocols that prevented modern DNA from contaminating the ancient samples. The genetic mapping process revealed something completely unexpected about the specimen's biological origins. Instead of the 70% human DNA match reported in 2023, the new analysis showed 99.7% human genetic sequences across all major chromosomal regions. The remaining 0.3% consisted of bacterial DNA from soil microorganisms that had colonized the specimens during their burial period, rather than any unknown or extraterrestrial genetic material. But the human DNA contained unusual patterns that explained the specimen's distinctive anatomical features. The genetic analysis identified specific mutations in the TBXD gene, which controls vertebral development, and the GLI3 gene, responsible for digit formation during embryonic development. These mutations occurred naturally in isolated populations, but were extremely rare in modern human populations, appearing in fewer than 1 in 100,000 individuals. The TBXD gene mutations explain the elongated spine structure and additional vertebrae found in both specimens. Normal human development produces 33 vertebrae, but these specimens displayed 37 vertebrae due to altered genetic expression during embryonic development. This condition, known as supernumerary vertebrae, creates the extended torso proportions that made the specimens appear non-human during initial examination. More significantly, the GLI3 gene mutations accounted for the three-fingered hand structure that had puzzled researchers since the discovery. This gene regulates the sonic hedgehog signaling pathway that determines finger number and formation during fetal development. The specific mutations identified in these specimens caused polysyndactyly, a condition where normal five-finger development reduces to 3 digits through fusion and regression of finger buds during embryonic growth. The DNA analysis also revealed genetic markers indicating these individuals belonged to a specific population group that lived in the Nazca region between 900 and 1100 CE. Mitochondrial DNA sequences matched haplogroups found in pre-Columbian South American populations, with the closest modern genetic relatives being indigenous communities from the central Andes Mountains. Y chromosome analysis of the male specimen showed paternal lineages that traced back to the earliest human migrations into South America approximately 15,000 years ago. Multiple independent laboratories confirmed these findings using different sequencing platforms and analytical approaches. The Max Planck Institute for Evolutionary Anthropology in Germany processed samples using their established ancient DNA protocols, while the University of Copenhagen's Center for Geogenetics analyzed specimens using their specialized techniques for degraded genetic material. Both teams produced identical results regarding the human origin and specific genetic mutations present in the specimens. The contamination issues that plagued the 2023 analysis were completely resolved through improved extraction protocols and environmental controls. Each laboratory maintained separate processing areas for ancient and modern DNA work, with different ventilation systems and equipment to prevent cross-contamination. Authentication tests confirmed that no modern human DNA had contaminated the ancient samples during processing or storage. Researchers also performed amino acid racemization tests on the extracted proteins to verify the age of the genetic material. This process measures the conversion rate of amino acids from their original forms to mirror image structures over time, providing an independent age verification method. The results confirm that the DNA originated from specimens that died approximately 1,000 years ago, consistent with the carbon dating estimates. The genetic evidence explained how environmental and cultural factors contributed to the specimen's unusual appearance. Population genetics analysis revealed that these individuals came from a small, isolated community with limited genetic diversity. Inbreeding within this population increased the likelihood of rare genetic mutations becoming fixed within the group, leading to the distinctive anatomical features observed in multiple individuals. DNA methylation patterns provided additional insights into the specimen's development and health status. These chemical modifications to DNA sequences indicated that both individuals had experienced nutritional stress during childhood, which affected their growth patterns and final adult size. The combination of genetic mutations and environmental factors created the unique physical characteristics that initially suggested non-human origins. Nuclear DNA analysis revealed that the two specimens were not closely related, despite sharing similar anatomical features. They came from the same population group, but were separated by at least 3 generations based on genetic distance calculations. This finding indicated that the unusual physical traits were relatively common within their community, rather than representing isolated mutations in a single family line. The comprehensive genetic analysis also identified several other inherited conditions present in the specimens. Both individuals carried mutations associated with dwarfism, specifically in the FGFR3 gene that regulates bone growth. Combined with the spinal and digital mutations, these genetic factors explained why fully adult individuals appeared so small and anatomically unusual compared to typical human remains from the same time period. The genetic evidence painted a clear picture of these specimens as members of a small, isolated human population that developed unique physical characteristics through a combination of rare genetic mutations and environmental pressures. However, the DNA analysis only told part of the story. To fully understand these mysterious beings, scientists needed to examine their internal structures with unprecedented precision using advanced imaging technology that could reveal secrets hidden beneath the surface. Advanced imaging technology reveals hidden secrets. Synchrotron-based MicroCT technology became accessible to forensic researchers in early 2025, revolutionizing how scientists could examine ancient remains. Unlike standard CT scanners used in 2023, these machines generated X-ray beams 10 billion times brighter than hospital equipment, capturing details as small as 0.5 micrometers. The European Synchrotron Radiation Facility in France processed both specimens using their most advanced beam lens, creating three-dimensional models with resolution previously impossible to achieve. The MicroCT scanning process required 72 continuous hours for each specimen, generating over 50,000 individual cross-sectional images. These slices were assembled using artificial intelligence algorithms that identified and separated different tissue types, bone densities, and foreign materials within the specimens. The resulting 3D reconstructions revealed internal structures that standard medical imaging had completely missed during the original investigation. Enhanced scanning immediately detected structural anomalies within the bone composition that explained the specimen's unusual appearance. The elongated skulls showed evidence of craniosynostosis, a condition where skull sutures fuse prematurely during childhood development. This medical condition creates the distinctive head shape by forcing brain growth in specific directions when normal expansion paths are blocked. The MicroCT revealed that both specimens had experienced this condition, but the suture fusion patterns were consistent with genetic causes rather than artificial skull binding practiced by some ancient cultures. Bone density analysis uncovered surprising information about the specimen's development and health status. The skeletal material showed density values approximately 15% lower than typical adult human bones, indicating chronic malnutrition during the growth period. However, the bone microstructure displayed normal trabecular patterns and osteocyte distribution, proving these were genuine biological bones that had developed naturally over years rather than artificial constructions made from carved materials or assembled from multiple sources. The three-fingered hand structure became much clearer through detailed internal imaging of the digit bones and joints. The MicroCT revealed that these hands had developed through normal embryonic processes, but genetic mutations had caused the fourth and fifth finger buds to fuse with the middle finger during fetal development. The bone fusion points showed seamless integration with no evidence of surgical modification or post-mortem manipulation. Joint surfaces displayed normal wear patterns consistent with decades of use, proving these individuals had lived full lives using their three-fingered hands for daily activities. Internal organ arrangements showed significant deviations from typical human anatomy, but these differences had clear biological explanations. The specimen's torsos contained additional rib pairs that created enlarged chest cavities to accommodate the elongated spine structure. The heart positioning had shifted slightly to the left to fit within the modified rib cage, while lung capacity appeared reduced due to the compressed thoracic space. These anatomical adaptations were consistent with the genetic mutations identified in the DNA analysis. The mysterious egg-like structures received particular attention during the advanced imaging process. High-resolution scans revealed that these objects were calcified organs rather than reproductive eggs. The specimens had developed kidney stones and gallstones over their lifetimes, with the mineral deposits growing to unusual sizes due to their specific diet and genetic predisposition. The positioning within the abdominal cavity matched the location of these organs in normal human anatomy, completely explaining the enigmatic oval shapes that had suggested alien reproduction methods. Isotope analysis of bone samples provided crucial geographic and dietary information about the specimen's origins. The scanning equipment could analyze trace elements within the bone matrix without destroying significant portions of the specimens. Strontium isotope ratios matched geological signatures from the central Andes region, specifically areas around the Nazca Plateau where the specimens were allegedly discovered. Lead isotope patterns indicated exposure to specific mineral sources found in high-altitude copper deposits common in that region. Metallurgical analysis detected unusual elements within the specimens, but these materials had straightforward explanations related to preservation and environmental exposure. High concentrations of copper, silver, and gold particles were embedded throughout the bone structure, consistent with burial in soil containing ore deposits from local mining activities. The Nazca region has extensive mineral resources that would naturally contaminate organic remains over centuries of ground contact. The advanced scans uncovered clear evidence of deliberate preservation techniques that explain the specimen's remarkable state of conservation. The bone surfaces showed microscopic traces of plant-based resins and mineral salts that had been applied as preservatives. These substances matched compounds used by pre-Columbian cultures for mummifying important individuals, including coca leaf extracts, quinoa-based adhesives, and locally sourced salt deposits. 3D reconstruction models allowed researchers to digitally separate different tissue layers and examine the specimen's construction in unprecedented detail. The imaging proved conclusively that both bodies consisted of complete, articulated skeletons from single individuals rather than assembled parts from multiple sources. Bone growth patterns, joint wear, and mineral deposition showed consistent aging throughout each skeleton, indicating these were intact remains of two people who had lived to approximately 40–50 years of age. The scanning technology also revealed evidence of healed fractures and degenerative joint disease that provided insights into the specimen's life experiences. Both individuals had experienced significant physical stress, including compression fractures in the spine, consistent with carrying heavy loads over long distances. These injury patterns matched the lifestyle of high-altitude communities that relied on llama herding and mountain trade routes for survival. Digital reconstruction allowed scientists to restore the specimens to their probable appearance at the time of death, before the preservation process altered their features. The models showed that these individuals would have appeared unusual but recognizably human when alive, with their distinctive characteristics resulting from rare genetic conditions rather than extraterrestrial origins. The imaging technology had provided definitive evidence to understand these mysterious specimens as remarkable examples of human biological diversity. However, the scientific community demanded more than just technological analysis to reach a final conclusion. The evidence required evaluation by independent experts who could assess these findings through the lens of decades of forensic experience and comparative knowledge. The forensic expert testimonies that changed everything. The International Forensic Examination Team, assembled in March 2024, represented decades of combined expertise across multiple scientific disciplines. Dr. Elena Rodriguez from the University of Barcelona brought 30 years of experience in forensic pathology, specializing in ancient remains and mummification techniques. Her work had previously identified fraud in several high-profile archaeological cases throughout Europe and South America. Dr. Michael Thompson from Johns Hopkins Medical School served as the lead radiologist, with extensive background in medical imaging of unusual skeletal conditions and genetic anomalies. The anthropological analysis was headed by Dr. Sarah Kim from the Smithsonian Institution, whose research focused specifically on pre-Columbian populations and cranial modifications in ancient cultures. These experts approached the specimens with completely different methodologies than the rushed 2023 examinations. Instead of single-day assessments, they conducted a 6-month investigation using standardized forensic protocols developed for analyzing disputed remains. Each expert worked independently initially, forming preliminary conclusions without consulting the others to prevent bias from influencing their findings. This approach contrasted sharply with the original presentations, where conclusions were announced publicly before completing comprehensive analysis. Dr. Rodriguez began her pathological examination by documenting every visible surface feature using high-resolution photography and detailed measurements. She identified multiple healed fractures throughout both skeletons, including compression fractures in the lumbar vertebrae and hairline cracks in the femur bones. These injury patterns suggested individuals who had performed heavy physical labor throughout their lives, consistent with high-altitude mountain populations that relied on manual labor for survival. The bone healing patterns showed normal human repair processes over years of recovery time. The radiological analysis conducted by Dr. Thompson revealed crucial details about the internal bone structure that previous imaging had missed. Using advanced CT reconstruction techniques, he mapped the complete skeletal architecture and identified specific growth patterns within the bone matrix. The three-fingered hands showed clear evidence of developmental fusion rather than surgical modification. Each finger contained the proper number of phalanges and joints, but genetic factors had caused adjacent digits to merge during embryonic development. The bone fusion points displayed seamless integration that would be impossible to achieve through artificial manipulation. Dr. Kim's anthropological assessment focused on comparing the skull formations to documented cases of cranial modification throughout South American cultures. She found that the elongated skull shape matched patterns seen in populations practicing artificial cranial deformation, a cultural practice where infants' heads were bound to create specific shapes considered aesthetically desirable. However, detailed examination revealed that these specimens showed natural craniosynostosis rather than artificial binding. The suture fusion patterns occurred due to genetic factors rather than external pressure during childhood development.